So what were we doing last time?
We talked about spermiogenesis, which turns spermatids into sperm, and starts with the construction of the acrosomal vesicle from the Golgi. And then the flagella forms from the centrioles, and the nucleus rotates so that the flagella is directed towards and into the lumen. And then chromatins commence incomplete shutdown transcription, and then residual body are left behind when all the cytoplasm leaves, and it's just like little bumps in the lumen. And then we talked about.
Okay, that's fine. And the residual bodies will be reabsorbed. They will essentially die. I mean, there's no nucleus. It's kind of cytoplasm. It will reabsorb. Okay.
Then we started oogenesis, which is activated by multiple signals, including hormones, paraffin factors, and just tissue anatomy. When we're an embryo, we have about 1,000 oogonia, and first 2 to 5 months, those rapidly divide until they reach about 7 million. And then they start dying until birth when there's about 2 million left. The surviving oogonia then undergo meiosis with increased STRA8 levels and become primary oocytes and get stuck at prophase 1, which is the diploatine stage, or the dictyate resting stage.
Yeah. Okay.
So then at puberty, the luteinizing hormone, is that right? Initiates meiosis, and then the first meiotic division happens, which cell makes 2 cells, and then the second meiotic division starts and is arrested at the second meiotic metaphase, and then ovulation occurs and fertilization will initiate the completion of the second meiotic division. And then we talked about meiotic regulation of the developing oocyte is regulated by the anatomy of the ovary. So then we drew like the ovary with the primordial follicles, and then we looked closer at the primordial follicles and how there is loosely arranged thecal cells around it, and then the granulose cells. And then at puberty, follicle stimulating hormone, FSH, is released, which causes a group of primordial follicles, about 5 to 9, to start growing. And then FSH induces 500-fold increase in the size of the oocyte. And we talked about how the granulous cell starts hitting GDF9, member of the TGF beta growth family, and then how that acts as a paraffin factor, and the granulous cells start dividing.
Okay, then what happened?
They divide and start forming the layers, and then the thecal cells are still there. But the follicular growth of FSD.
Follicular stimulating.
Yes, sorry. Induces an expression of luteinizing hormone receptors on the adenosine granulosa cells. Then we talked about mutations in the GDF9, which causes premature ovarian failure, which loses 2 oocytes at the same time, which is why we have the different things. And then we talked about the fully grown oocyte, and then we have the extracellular, we have the oocyte, the extracellular matrix is Cynthia, and then the granulose cells divided crazy, and a cavity is created called the atrium.
Antrum.
Antrum, sorry.
Yeah. Okay, then what happened?
The antrum changed the nature of proteins and hormones. And then we talked about if we took the oocyte out of its basically surroundings, and whether we had only the oocyte plus the cutinous layer, which is the 3 to 4 layers surrounding the corona radiata, they will immediately start meiosis, which tells us that the granulosa cells contain some type of inhibitory signal preventing meiosis.
Okay. So what was the last thing you wrote? I think you
The last
provided
thing I
me
wrote
something
was,
wrong there. No, before that, the rupture thing. What was?
Oh, yeah, sorry. we talked about how,
Uh-huh.
where-
Graphene follicle is
Yeah.
there, okay.
And then there's a signal that comes down through the granulosa cells into the zona molecula, and then it like ruptures.
No, it's not to rupture.
Okay.
That's what I read in here. It's not to rupture. We were saying that if you take out, we said this graphene follicle is formed, right?
Yeah.
If you take out, if you open it yourself and you take out the oocyte with cumulus cell or oocyte only, then this cell will start going to the phase of meiosis. We didn't say that it sent a signal to rupture,
Okay.
right? That we didn't talk at all there for the rupture thing. We just said that outer cells, they send the signal that control the meiosis. The signal are coming from the outer cell to control the meiosis, not the rupture.
Gotcha.
Rupture is breakdown, right? Taking them out. Meiosis is going through the cell wall. Okay, so that was wrong in your, which you sent me, that's why I can't. Okay, then, any question here?
Yeah, so I just had a question. When we talked about taking like the oocyte out or the oocyte with the cumulus,
Mhm.
does the antium come with?
No.
Okay.
Antrum is just a cavity, there is nothing, right? We're just talking about the cells surrounding it. Those are cumulus, right? So in cumulus, we don't have any cavity, right? So, okay. So we were saying that what is happening? We were saying that, so let me draw it a little bit again. So these are the outer granular cell, right? We were saying that outer granular cells, these cells make cyclic GMP. And by the way, I have actually put the total subtopic list on the Moodle so that you can actually, if you're writing it, you can basically follow it, right? So, cyclic GMP is made here. Cyclic GMP pass through these gap junctions. There are gap junctions between one cell, next cell like this, and then ultimately it reaches the oocyte. And this oocyte is primary oocyte, right? Actually, it's not still primary, it's still the oocyte. Still the oocyte. Though it has not divided. It's just a oocyte, right? And then, so cyclic GMP is coming from here, cyclic GMP is reaching the oocyte. Okay. So what cyclic GMP is doing? We are saying that cyclic GMP it inhibit phosphodiesterase. It inhibit phosphodiesterase. And so, cyclic GMP is reaching here, right here is phosphodiesterase enzyme, right? And that is inhibited. And what phosphodiesterase does? So phosphodiesterase is an enzyme. What it does, it degrades cyclic AMP. It degrades cyclic AMP. And so phosphodiesterase reach here, PD reach here, PD blocks cyclic AMP. Okay? Not block, rather kind of destroy it. Destroy it, degrade it, blip, okay? And then what cyclic AMP does, cyclic AMP inhibits progression to meiosis. So, cyclic AMP what does, it basically inhibit migration to meiosis. So now think about it. if the cyclic GMP is coming there, we said outer cells have something, right? That does not allow these oocyte to undergo meiosis, right? We said if you take out this plus all those cells, this cell will undergo meiosis. Now, why it will undergo meiosis? Because cyclic GMP is coming, cyclic GMP blocking phosphodiesterase. If there is phosphodiesterase, what phosphodiesterase will do? It will degrade cyclic AMP, and what cyclic AMP was doing? Inhibiting meiosis. Making sense? So that inhibition is gone. So cyclic AMP will be degraded by the enzyme phosphodiesterase, and the meiosis will take place. Making sense? So what cyclic AMP does actually? So cyclic AMP, if you remember from your cell signaling, from the cell biology, so this cyclic AMP, what it does, it essentially activates protein kinase C, PKC, right? So protein kinase C is active, then it also activates PKA, protein kinase A. So in this particular aspect, we are more related to PKA. So forget about this. So activate PKA, okay, protein kinase A. Protein kinase A. So when the protein kinase A is active, what it does? It activates V1. If you remember, what is V1? V1 is the protein that inhibits mitosis promoting factor. So V1 activation, what it does, that inhibits mitosis promoting factor. So I'm writing here M or MPF. So these factors, we call it mitosis promoting factor, but these factors are responsible to initiate the cell division, be it meiosis or be it mitosis. Okay? So that activate V1, and V1 is going to inhibit mitosis promoting factor. Then it also, because this is a kinase, what is the purpose of kinase? Kinase phosphorylate the substrate. So phosphorylation of V1 activates it, right? Other than what it does, second thing, it phosphorylates CDC25. So two things it's doing. Cyclic AMP is activating PKA. PKA is doing two things. Number one, activate V1. Activation of V1 inhibit mitosis promoting factor. And actually the CDC25 is a protein that actually activate MPF. If you remember, I don't know how much cell signaling you have done, but remember that phosphorylation for certain protein can activate it, phosphorylation for certain protein can deactivate it. So CDC5, when it is phosphorylated, it is actually deactivated. So if it is deactivated, what it can do, then what will do, then if it is deactivated, then it cannot activate NPF. So two things are happening here, two pathways. But both the pathways, what they are doing, both the pathways are inhibiting mitosis promoting factor. So going back, we are saying cyclic AMP inhibit the progression to meiosis. It does that because it activates PKA. When the PKA is active, it's a kinase, okay? That PKA activate V1. Once it activates V1, V1 activity inhibit mitosis promoting factor, so it cannot undergo cell division. Now normally cell division is activated by CDC25 once it is activated. But what pKa does, pKa phosphorylates CDC25, and when it is phosphorylated, it cannot activate mitosis promoting factor. So in both ways, the meiosis is blocked. That is making sense? So if you had a way to take out these cells away, if these cells are away, then what is going to happen? Phosphodiesterase will be active. Phosphodiesterase active, it will degrade CMP. Then pKa will not be active. Then V will not be active. Then this inhibition will go. Right? And then if pKa is not active, CDC will not be phosphorylated, so CDC5 then can activate NPL. Making sense? Okay, then what happened? So we said in the beginning when you were reading, we said this is made in dictyate stage, right? And we also said that in dictyate stage, follicular stimulating hormone come out, right? And what follicular stimulating hormone does, two things it was doing. It was increasing the size, right? And the second thing it was doing, it was making right here the receptors. What were those receptors? Luteinizing hormone receptor. So those are luteinizing hormone receptors. Okay. So what happened? We said this stays like this till puberty, right? So they are in dictyate stage till puberty, right? So they are now in diplotin stage because you are having cyclic GMP coming here, right? So how to move forward? That means you have to somehow get rid of this cyclic GMP if you want it to undergo meiosis. So then at puberty, we said the meiosis starts again. So at puberty, what happened? The luteinizing hormone it is made up, its level goes up at puberty, right? And then, so luteinizing hormone is coming here, it binds to its receptors. So luteinizing hormone is coming here and binds to its receptor. So once it binds, it does two things. So bind to receptor, it means that signaling cascade will activate in these cells, right? So luteinizing hormone, so LH receptors will be activated. Okay, so it means you will have signal transduction. In where signal transduction will be in the outer granulosa cell, right? In outer granulosa cells. So these outer granulosa, outer follicular cell will have that signaling there. And what that signaling is going to do, that signaling is going to do two things. Number one, that is going to, so two effects are going to happen. Number one will be right here at this place. Gap junctions will be closed. So gap junctions will be closed. Okay? And then, second thing, the production of cGMP will be going down. So if cGMP is going down, gap junctions are closed, so cyclic GMP cannot reach here, right? So therefore, cGMP cannot reach oocyte. Okay? So if cGMP cannot reach oocyte, what is going to happen? Therefore, cyclic AMP will be degraded. That is making sense? So now if the cyclic GMP is degraded, then what is going to happen? So this oocyte is there, cyclic GMP is degraded. So therefore, MPF, mitosis promoting factor, will be activated. Okay? And if NPF is activated, then what is going to happen? We are going to say that meiosis is going to start, right? So meiosis start. I will say resume because it started early. Confusing?
If it's a mitosis?
Meiosis.
But-
Yeah, we are saying mitosis promoting factors. This is basically called mitosis promoting factor. It's the same protein that work for both meiosis as well as mitosis. Those are the cell cycle proteins, so we don't change their name. So, the meiosis which begin early is going to resume. That block will be gone, right? So if that block is gone, then what is going to happen? So you should have spindle formation there, right? So spindle formation is essentially myosin interacting with actin will initiate spindle formation. Right? So if that, and spindle formation will be near the cortex, not in the center, that will be near the cortex. So spindle formation is going to take place right here near the cortex. Because remember, this is the cortical side of that, right? And then what is going to happen? The first meiotic division will be completed. First meiotic division will be completed, okay? And that will lead to beginning of second meiotic division. So what we are saying? We are saying, hey, during this process, orthogranular cells are there, or outer follicular cells are there, they make cyclic GMP. Cyclic GMP, through gap junctions, crosses all those cells, goes to cumulus cell, and ultimately reaches oocyte. Once it reaches oocyte, it blocks phosphodiesterase, right? The purpose of phosphodiesterase is to block the cyclic AMP. So if the cyclic AMP is there, what cyclic AMP does? Cyclic AMP inhibits progression of meiosis by activation of pKa. pKa phosphorylate B1 and CDC25. Phosphorylation of CDC25 means it cannot activate MPF, and phosphorylation of B1 means it inhibits MPF. These are called mitosis promoting factor because these are cell cycle protein. But here they are going to induce the meiosis, okay? So what happens? So they stay there till puberty. At puberty, you have surge of luteinizing hormone. That luteinizing hormone binds to the receptors of luteinizing hormone receptor. Remember, those are only present on the outer granulosa or outer follicular cell. Once it binds there, it starts a signaling cascade in these cells, and what that does, that closes the gap junction and decreases the function of production of cyclic GMP. So now the cyclic GMP cannot come here. That means now cyclic GMP is not there, so means phosphodiesterase is active. If phosphodiesterase is active, that's going to degrade cyclic AMP. If now cyclic AMP is degraded, so then MPF now can be activated. Once it activates, that initiate the meiosis, actually restart the meiosis. And then the spindle formation takes place, that is through the interaction of myosin with the actin, and that spindle formation will take place right here near the periphery in the cortical cytoplasm. First division is going to start. And then the first division is going to start, first division is finished. Then what is going to happen? Second division start. And when the second division starts, it reaches up to meiotic second, it reaches up to second meiotic metaphase. It reaches up to there. And then there's a factor called, that is present right here, made right here in the oocyte, called cytostatic factor. A factor called cytostatic factor comes there. And what cytostatic factor does, it binds to MPF. And when it binds to MPF, it inhibits MPF. So MPF is now inhibited. Now what happened at this stage, when the MPF is inhibited, that means division is stopped at second meiotic metaphase. At this stage, ovulation occurs. At this stage, ovulation occurs. Okay?
So before MPF is bound, or?
No, MPF is blocked after that, because you stop it in the second meiotic metaphase. So now it's stopped there. So now the ovulation will occur. If fertilization occurs, then it will move forward. If it does not, it will just die. Okay? So what happened in the beginning? We said a group of primordial follicles, they start growing. We said 5 to 7. Right? So out of 5 to 7, all will die except one will reach this stage. So we will not have 5 or 7 eggs ovulated at one time. Only one is ovulated. So that means in that whole group, only one oocyte will reach to this stage. Now, when the first division takes place, then it becomes primary oocyte. I will talk later, explain that, how that happens, right? So the primary oocyte, so only, just remember, out of group of 5 to 7, only one is going to survive to reach this stage for the ovulation. Okay? So talk about these processes we just talked, then we'll go further and explain what is going to happen next.
So the implementation of the GDF gives us like 2 oocytes, is that just like, they don't all die, or is that really something another set? Yeah.
So the oocytes would degrade C and C. Which closes the gap junctions and stops the production of CG and B is decreased. And since CGMP can't reach that site, the CAMP will be degraded and then MPF is activated and meiosis resuming, and then the myosin interacting with actin will initiate spindle formation near cortex. And then first meiotic division is complete. And the second meiosis, and then it reaches up to the second meiosis, and then factor 9 to MPF. And then so ovulation occurs before
It's like we're up here, so whatever, but it stops at the second meiotic phase. So like, when it's at that, I guess during the second meiotic phase. Like, not ovulation. Yeah, because ovulation occurs in the last, and then it's fertilized in the second. Baby. So you said only one of the oocytes out of like the 5 to 9 will reach ovulation, but during
Yes.
this mutation we get 2 oocytes through? Is that like?
Yes, in the
two
mutation,
mutations?
remember that the mutation is happening, this process is not occurring properly, right?
Yeah,
It's a premature thing.
okay.
So that's why we call it ovarian failure, because the ovary cannot function properly. So you can have 2, 3, but usually it is 2.
Is it 2 to 3 groups of the 5 to 9 cells, or is it just
No, no,
like
no,
2 of the cells don't die out?
So what is going to happen? So when this process is not working, it means there is something bad about this thing, right? It means those cells which were surrounding it are not dividing, right? So when they are not dividing, ovarian failure takes place. Ovarian failure is usually the case that is telling you, hey, something is messed up, let's kind of like wrap up the things. And during that wrap up, what is going to happen, most of those cells will die. But occasionally, 1 or 2 will survive, but it will not be like fully mature. But remember, if these cells are not dividing, they are not producing any luteinizing hormone receptor. That means they can undergo division, right? And when they are not there, it's easy for them to rupture and they will be thrown out of the ovary more than 1, 2. And that's why you said that you can have fraternal twins. So instead of 1, usually when it's controlled, you have all other die, only 1 survive, 1. But when they have not reached up to that process where it should be like this, right?
Mm-hmm.
So it means it's all uncontrolled. So system is not working, you can have more than 1.
Gotcha.
Yeah. Question here? Anything here which is not clear? You're okay? Okay, now. What is happening? So we are saying that now the puberty is there, right? So luteinizing hormone has done what it was supposed to do, put it back into meiosis. And now we are saying that cytostatic factor is there. Now the ovulation need to take place, right? So in ovulation, what is happening, for example, this is your ovary, and I will show you a video in a second. This is your ovary, and right here is your this is the oviduct. In common language, this is also called fallopian tube. Okay. So these finger-like projection are there. These are called fimbriae. I'm not exactly sure about the spellings, but fimbriae. Okay. So these are finger-like projection. So in ovulation, what is going to happen? From here, from here, from this ovary. So remember, we had like many Graafian, follicle, primordial follicle maturing, one becoming Graafian follicle that ruptured, right? That is ruptured, it is going to send that oocyte here. That will be called ovulation, right? So that oocyte, now this is actually a primary oocyte because undergo the first meiotic division. Primary oocyte will be captured by these fimbriae. These fimbriae are going to catch it, and this is going to then move here in this area, and it will be surrounded by all the cumulus cells. That is going to happen. Okay? Here, right here somewhere in this area we call infundibulum. In this area the fertilization is going to take place, right? And this area, this is the uterus area. So what is going to happen? So we are saying that you have at puberty, luteinizing hormone is very high. That luteinizing hormone has initiated the process of meiosis, right? But that luteinizing hormone also initiates ovulation. So ovulation is the process of oocyte coming out from the Graafian follicle and then captured by the oviduct. Okay? Now, what is happening? What luteinizing hormone does? Luteinizing hormone activates the production of progesterone. Progesterone is another hormone. That hormone is made by gonads as well as made by the adrenal cortex, adrenal gland. Okay? So progesterone made in gonads, made by gonads and adrenal gland. These are the two sources. Okay. Now what happened? So if hormone is there should be receptors, right? So in this ovary system, ovary is making now progesterone because the luteinizing hormone is there, right? So the receptors for progesterone are present on the cells, on the follicular cells. So remember, this is your Graafian follicle, right here. All these cells are there. Right, you have all the cells there. Right here is your, now it is oocyte, because it has undergone the first division, right? It's a primary oocyte now. So this is your oocyte, surrounded by zona pellucida. Then you have all the cells there. You can say cumulus cells are there. All these cumulus cells are there. Right here is your cavity enterum. Okay? So the receptors of these are right here on these cells, right? So now, if you want this thing to come out of this, you need to rupture it, right? You need to break it. So to break it, what you have to do, you have to make, how to break the cell from cell? That means you have to break the cell adhesion protein, right? So the cell to cell, they are joined by each other by what you call cell adhesion protein. Cell adhesion proteins. So these proteins which are here, they should be removed, right? So they can be removed by the enzymes, and those enzymes, what you call proteases, So proteases level should go up. You should make more proteases in these cells. And you make those proteases there, so those proteases then can remove these cells, so cell can separate from each other. Right? So the receptors for these progesterone are present on these cells right here. All these cells have the receptors. Okay? So what happened? Luteinizing hormone is there. Luteinizing hormone coming to these receptors, and what is going to happen? They are going to make, so they do two things. Luteinizing hormone activate progesterone. Right here is progesterone. Progesterone does two things. Number one, it going to activate genes, those make proteases. Okay? So the proteases are being made. So if the proteases are being made, what is going to happen? These cell adhesion protein will be disrupted, and if these are going to be disrupted, this thing will open. So what happened? This thing opens up like this. Okay? Now this thing is now free, this thing will open up because these were making, but these are not making, right? Only outer group of cells are making. Now this need to come out. It's in. So that means you have to do some sort of muscle contraction, right? So that it can come out. So it means the first thing, it should activate gene, those make proteases. Second thing is, it should activate genes for muscle contraction. So gene for muscle contraction are activated. And when that happens, this thing, so these cells are left there, whatever is there. But out of this, you have this oocyte, and here are, so this is oocyte plus cumulus is out. Making sense? Yes.
So, does the LH bind or does the progesterone bind to cause those two?
Luteinizing hormone produces progesterone. Progesterone binds to its receptor, which are expressed on the cells of the follicle, right? So then progesterone activates cell signaling pathways, and how progesterone is a hormone, right? So it can directly activate the gene expression. So it activates the gene expression for proteases. Once proteases are activated, so these cells, their cell adhesion proteins are disintegrated, it opens up, right? Then what you need to do is propel it out. How to propel it out? You cannot go and kick. You need to have muscle contraction, right? So then you need to activate the more gene which can make more muscle contraction protein. So then muscle contraction start, and then this oocyte with cumulus come out of this graphene follicle. So far it is okay?
Is all of this happening in the ovaries though?
Yes, everything is happening in the ovary. So now it comes out. It comes then out of the ovary, it will come out of the ovary and will be catched by these fibrils. Okay? Now what is happening there? This remaining portion which is there, this remaining portion, this portion, which is, so now this oocyte plus cumulus is out, right? So this remaining portion which is there, this is called corpus luteum. This is called corpus luteum. This is called corpus luteum. Okay. Now, what this corpus luteum does, it actually makes huge amount of progesterone. Huge amount of progesterone. it makes huge amount of progesterone. What progesterone does? Huge amount. So that progesterone is made. So it does two things. Number one, it binds to its receptors in uterus. Hormones are released in the blood, right? So hormone is reaching there. So hormone reaches here in the uterus. So in the uterus, the cells here, they have the receptors. So bind to receptor in the cells of uterus. And what it does, once it binds there, it keeps uterus in the stage. So you have to keep in the stage so that it can maintain pregnancy, right? The stage to maintain pregnancy. Other thing it does, that's about the first thing. Second thing it does, inhibit production of luteinizing hormone. So then luteinizing hormone go down. Remember, if luteinizing hormone stay up, it's going to activate, come here, start growth of one more follicle, one more ovulation, right? Therefore, blocks further ovulation. So that's why when pregnancy is occurring, you have only one embryo developing, otherwise you have 10, 20, 50 developing here, right? Because you want to stop the ovulation. And all the birth control pills, they have progesterone. And that's the purpose, to inhibit the luteinizing hormone so that you can inhibit the ovulation. Okay? So now, let's go back. We are saying that the oocyte, primary oocyte is formed because you had the luteinizing hormone that basically act on the receptor, block the entry of cyclic GMP. Then we said you had the mature Griffin follicle that is now ready to ovulate, right? And we are saying that also happened because of luteinizing hormone. Because luteinizing hormone, it induces the production of progesterone. Now the progesterone is made. What progesterone does, it binds to receptor here, and when it binds there, it activates the genes for proteases. They are going to break the cell adhesion protein so that the ovum with the cumulus cell can be released out. And then we said it also activates the genes for muscle contraction so that oocyte can be released from here and can give the pulse here, right? And we said when this oocyte with cumulus is released, the rest of the portion is called corpus luteum. And corpus luteum then make a huge amount of progesterone. That progesterone reaches out in the area of uterus. Its receptors are there in those cells. It tells it to maintain the state of pregnancy. And then it binds to the genes which are making luteinizing hormone, inhibit the production of luteinizing hormone so that no more ovulation can take place. And that's why it is also in the birth control pills. Now let me show you this movie, what is happening. Remember, this is a kind of a movie they're just showing, they don't have the name of the hormones, right? So just discard that. But look at the process, okay?
Immature egg at its center. The typical 28-day menstrual cycle begins on the first day of menstrual bleeding. During the first 7 days of the cycle, a few follicles start to grow. These maturing follicles secrete estrogen hormone into the bloodstream to prepare the lining of the uterus for pregnancy. Around day 7, all of the follicles stop growing and begin to degenerate, except for one. This dominant follicle continues to grow, nourishing the developing egg inside it. Around day 12, the follicle secretes a surge of estrogen into the bloodstream. When the estrogen reaches the hypothalamus and the pituitary gland in the brain. The anterior or front part of the pituitary gland floods the bloodstream with luteinizing hormone. Around day 14, luteinizing hormone causes the follicle to undergo a sudden growth spurt. Right before ovulation, the egg detaches from the inside of the follicle. The bulging follicle releases chemicals, signaling one of the two fallopian tubes to move in closer and envelop the follicle. The follicle swells until it bursts open, ejecting the ovum and fluid from the follicle into the abdominal cavity. In response, the fimbriae, tiny finger-like projections at the end of the fallopian tube, sweep across the ovulation site and pick up the ovum. Microscopic cilia on the fimbrial surface guide the ovum to the entrance of the fallopian tube. Muscular contractions along the wall of the fallopian tube gently propel the ovum toward the uterus.
Okay. So this was the process as it is. They didn't detail all the thing, so but detail we know, right? So just talk to each other for what we just, discussed, and then we'll go further.
Yeah, so the closer ones have the receptors, right? I think.
Confusing.
So the receptors that are present on the follicle.
Receptor for what?
For progesterone.
Yes, they are present on the
follicle
follicle cells, yeah.
So is that
Th-
like,
That cause the activation of, that cause the production of proteases there, right?
right?
And then proteases are going to break the cell adhesion, right?
So
it
that's
will
more
open.
like on the outside then?
Yeah, that is mostly on the outside, not on the cumulus. Nothing is on the cumulus, everything surrounding that. Otherwise, cumulus will also burst, right?
Right.
Yeah. So then other receptors are present on the cells in the uterus.
Right, yeah.
Anything else that is not clear? Any other confusion?
I think we're
Good?
good. I think so.
Okay. Any confusion here? No? Okay, so then we want to talk about oogenic meiosis. So remember that first part of the meiosis, the first part begins in embryo, right? We said around 2 million are there at the time of birth. 2 million of oocyte or oogonia are at birth. And they will remain in dictyoid, and meiosis start, we know that. And they will remain in dictyoid stage maybe for 4 decades, many of them. Many will remain in dictyoid stage for almost 4 decades. But all will be in dictyoid for almost 12 years, right? Because the meiosis restart at puberty. Now out of those 2 million, actually only 400 will actually undergo meiosis in lifetime. Remember we said 5, 7 grow, many die, right? So this will be the case there. So when the oogenesis, when this meiosis takes place, okay? So in the first stage, what is going to happen? We said the meiosis reinitiate when cyclic AMP is degraded, right? And we said that all of the spindle, they will move in the periphery, in the cortical periphery, right? And when they are going to reach here, they are going to arrange like this. So a group of chromosome here, group of chromosome here. Usually when the cell divide, you have spindle formation from pole to pole, right? So cell divide like this in half. But here this is not going to happen. They will arrange in the periphery, and that is going to lead to a cell production like this, a big cell and a small cell. Okay. So this large cell which is produced here, so this is primary oocyte, this will be called polar body. It will be called first polar body. And within 21 hours, this first polar body degenerate. First polar body will be degenerate. Okay. So this larger cell will be then called secondary oocyte. And then again a division is going to take place same way. So you will have the nucleus right here somewhere. And this will not be a nucleus, kind of an open chromosome kind of like thing. Then the second division is going to take place. And when that is going to take place, again the same thing is going to happen. Again the spindle formation will take place at the edges, and you will have another cell produced like this. And you will have, this will be your second polar body. And this will also degenerate. And what you will get? You will get a ovum. Right here. Now you can see here what is happening. Most of the cytoplasm is preserved, right? Most of the cytoplasm is preserved right there. So, but what is happening in this case? Because it is staying for such a long period in dictite stage, that leads to loss of cohesion. Loss of cohesion. What is cohesion? We know about those. What is cohesion, Rachel?
That's the thing that holds the.
Yeah, those proteins, those keep the chromatid attached to each other. And when that is not, that loss of cohesion, what that is going to lead? That is going to lead to aneuploidy. So loss of cohesion, that is going to lead to aneuploidy. Aneuploidy means you don't have normal number of chromosomes, right? So that means incorrect number of chromosomes. So, that is actually so bad if you look here. So you will have the trisomy. Up to age of 22, the chances of trisomy are almost 2%. But after that, after 30, you can see there's exponential increase in the chances of trisomy. So that's the bad thing of having those things arrested in this stage for such a long time. And here you can see all this green which you are seeing here, that is the cohesion, and here all the cohesion is going down. So that leads to aneuploidy. By 40 is almost 35% chance of having trisomy present in the newborn babies. So that's why a few people suggest like it's better to become mom early so that these chances are not there. So then the last thing what happened, so that was about the oogenic meiosis. So I think we'll not be able to finish in that 2-3 minutes what we want to talk. Maybe let's tend to just stop it here. So we have to be there by 10:10 also. So any question here so far? So we'll be finishing in first 5 minutes this topic. We have just one small thing to talk about. And then we'll talk the next topic on